Thanks for doing this. Thanks for joining me today. And congrats again on this study. I believe it took 3 years. Is that right, for the research and to publication?
I guess it must be getting on for that, Robin, yes? About 3 years?
Yeah, more or less. From the time we decided to do the study, search for funds, collaborators, decide on the experimental plans, do the experiments, analyze the data. This is a long process.
A long process. So this study, as I look at it, it's examine the effects of glyphosate and a glyphosate product that's part of the Roundup brand developed by Monsanto. And you looked at the effects on the microbiome of in the gut of female rats. What did you conclude? Michael, maybe you want to tell us.
Yes. What we found was that there was that, first of all, the glyphosate, both the glyphosate and the Roundup did have an effect on gut bacterial population composition. Now, what do I mean by that? We know that our gut is inhabited by thousands of different types of bacteria, and a balance in their composition and, more importantly, in their function is crucial for our health. This is a key point. We've all heard how important balance of our microbiome is to our health these days, and with the surge in sales of probiotics that people are taking to help to do that. And so anything that negatively disturbs the gut microbiome in terms of either the population of the bacteria or their function, their biochemistry, or both, has the potential of causing ill health, because we go from balanced functioning that is conducive to health to imbalanced functioning that may lead to a whole spectrum of different diseases. What we found in...
Can you give us an example of what the gut microbiome relates to specifically in our health? I mean, can we be talking about cancer and other diseases as serious as cancer?
Yes, gut microbiome imbalance or dysbiosis, as it has been, as it's called, has been linked to many different conditions: liver dysfunction, cardiovascular dysfunction, behavioral problems such as ADHD or depression, and also to cancer as well. These links have been established. It's a developing field. It's a rapidly evolving, developing field. And so we don't know everything that's there yet, exactly what changes occur in the gut and would give rise to an illness. That link is not very well established, but we do know that there are correlates at the moment. I don't know if Robin wants to add anything more to that, but generally speaking, this is what we're looking at. The changes in the gut microbiome may be neutral, because the changes occur in our gut microbiome quite frequently, depending on the inputs that we have.
And when I read through the paper, it seems that, so you did find changes in the gut microbiome.
We did.
You found some things that look like they might be indicative of oxidative stress.
Yes.
You seem to maybe have countered a notion that's been out there that glyphosate might act as an antibiotic in the gut. Can you?
Yes, the changes in the population of bacteria were not very pronounced. Let's put it that we did see changes with both Roundup and the glyphosate. There was an overlap, and then there was one change that was specific to the Roundup treatment. But overall, the changes were not huge. They didn't affect vast numbers of different bacteria. But what was more striking, and this is really one of the key messages of our paper, what was more striking was that the biochemistry of the gut was altered more dramatically. So in other words, although the bacteria didn't change very much, their function, their biochemical function was changed. And this is this is an important observation that's increasingly being recognized in the field of gut microbiome biology, that just looking at changes in the bacterial population is not enough these days. If you really want to get insight into the impact of a chemical pollutant, for example, like we investigated in our study, you really need to look at the biochemistry. And we found some very dramatic changes, alterations in the gut biochemistry. Two things stood out. Firstly, that the biochemical pathway that glyphosate and Roundup inhibit in plants that causes the plants to die. In other words, the mechanism through which glyphosate acts as a weed killer, it caused exactly the same type of biochemical change in the gut bacteria as it does in plants. That was something that was predicted some time ago, and we are the first to actually prove that inhibition of the plant system that leads to the plant death also happens in bacteria that share the same biochemical pathway, what's known as the shikimate pathway. Now, the inhibition of the shikimate pathway probably didn't alter the gut composition very much. Maybe Robin will correct me on this. We think that the changes in the population of the gut bacteria were through other mechanisms. And that's because the diet of the consumer, in this case, the rats, is probably, we feel, compensating. The nutrients in the diet are compensating for the inhibition of the biochemistry caused by the glyphosate and the Roundup, akin to what happens in plants, inhibition of the shikimate pathway. But what more striking were the changes in the biochemistry composition of the gut, indicative of what is known as oxidative stress. Now, what is oxidative stress? Oxidative stress is basically damage or stress that's caused to excessive production of what we call reactive oxygen. We in our bodies all the time generate what are known as reactive oxygen species. These are oxygen-type molecules that, as the name implies, are highly reactive. So they will react and damage just about any type of structure in our bodies, proteins and DNA as well. Now, normally we have mechanisms of neutralizing this. We have antioxidant systems of dealing with this. And also ingestion of foods that are rich in antioxidants help us to cope with this naturally produced reactive oxygen molecules. But what we saw in the gut of the rats that fed both glyphosate and the Roundup, and especially the Roundup, actually was more pronounced in the Roundup fed animals, was that was it stimulated an excessive production of this reactive oxygen. And perhaps even more important, so in other words, the gut was under oxidative stress. You know, there was an insult there. Especially the Roundup, there was a chemical insult there that put it under oxidative stress. But when we also correlated that, or looked in addition to the blood biochemistry of these very same animals, we saw very clear markers of oxidative stress as well in the blood biochemistry. So here we're seeing oxidative stress in the gut, and we're seeing clear suggestion of oxidative stress in the internal body of the animals. Now, why is this worrying, you may ask? It's worrying because oxidative stress, as I mentioned already, damages structures of the cells, can damage organs, can damage cells, and it can also damage DNA.
This study doesn't take it to that step. Is that correct?
No, it doesn't.
Okay.
It's a prediction.
Okay.
We have a prediction here based especially on the changes in the blood biochemistry. It's suggestive of oxidative stress.
Correct.
That this, if we looked further, we may find damage to organs, organ structure, and also, perhaps more importantly, damage to DNA.
Right.
And damage to DNA is not a good thing. That's an established risk factor for cancer.
Sure, of course.
So yes, indeed, our study is crying out to be followed up. And I can say right now that we are following up this observation, and that we are in the middle of investigating to see whether indeed the prediction of DNA damage resulting from oxidative stress is actually taking place. And we hope to be in a position to report on the findings of this follow-up study in the not too distant future.
Wonderful, wonderful. And Robin, perhaps could you speak to the dosages here, the levels? It's my understanding that these, the glyphosate and the Roundup product that the rats were given, I believe, in drinking water, is that correct? In their drinking water, was at levels that European and US regulators consider safe?
Yeah, we used several doses because this is the best way to find patterns in toxicology. And we wanted to start by doses which are relevant for human consumption. So we took first the permitted daily intake of glyphosate. So this is 0.5 milligram per kilo. In fact, this represents the dose of glyphosate which is considered to be safe for humans who are exposed daily to glyphosate. So in theory, we can ingest this dose of glyphosate daily without any harm. And then we increased this dose to other higher dose, which are also considered as safe by European agencies and American agencies.
Okay, so these levels that the rats were consuming were at levels that in comparison humans would be exposed to in their daily diet, perhaps.
It's not necessarily the levels to which we are exposed, but this is the levels which are deemed as safe. The levels we are exposed to are actually much lower, and new studies will have to be done, especially in human population, to understand if there is an effect. And this is also why our study is very important, because with these markers in the gut microbiome, we discovered a specific signature of glyphosate effect. And you can measure this in human populations. And if the levels of this signature is correlating to glyphosate level, you can conclude that maybe there is an effect or not in humans.
Okay, so this study, your work here, doesn't necessarily speak to the issue that some people are concerned about glyphosate residues in food.
It's a first step. It's a first step, and we need a marker to understand if glyphosate has an effect, and we are providing this marker.
There have been other studies looking at the gut microbiome in animals, right? I believe there have been other studies in rats even in recent years. How is this one particularly novel, or how does this one particularly advance the science?
Yeah, this one is more advanced because we looked at a wide range of glyphosate effects, the composition and the function of the gut microbiome. Because the previous studies which were done, they only looked at the composition of the gut microbiome using a rather shallow method. They only measured a couple of bacteria and concluded based on this. What we did is to measure every piece of DNA, sequence every piece of DNA in the gut, so we can have a whole picture of the level of every microorganism in the gut, fungi, viruses, bacteria. And on top of that, we looked at the profiles in small molecules, what we call the metabolome. And this is the most important because this was never done, and this was where the effects of glyphosate were the most visible.
So I'm curious about why glyphosate, right? I mean, I've written a lot about it, obviously. It's the most widely used herbicide. It's been in the news because of litigation linking it to cancer, but there are also, numerous other pesticides out there, thousands. And, should we be looking at their impact on the gut microbiome as well?
Maybe Michael, you take that one. Or...
Sure.
This is a very good point, Carey. Indeed, I mean, glyphosate is something that my group, I've been working on this with Robin in my group now for a number of years. So it was and it's of interest to us, not historically, because Robin was working on glyphosate even before he joined me, some 6 years ago. But it's what you say about, and the reason why it's important is because it is so universally used all over the world. As you say, it's the most widely used weed killer in the world, and its exposure is almost ubiquitous, especially in heavy use nations such as the United States. We probably find higher residue levels of glyphosate in the urine of Americans than probably anywhere else in the world, because it's just used so widely in so many different agricultural systems, especially glyphosate tolerant GMO cropping systems. But you're right in what you say, that we shouldn't think that glyphosate is the only worry in the family of pesticides that may cause harm. Indeed, we're exposed to tens of different chemical pollutants every day, just through our food, but also, of course, in other aspects of our living environment. And so considering mixture effects is very, very crucial, because it actually reflects a real world situation, because we're not just exposed to one chemical, we're exposed to a mixture, which is why we have also taken to we have begun to investigate pesticide mixtures as well. We have a study that is currently has been submitted for publication, and we hope that will come out in the next few months, where we have administered to rats a mixture of 6 pesticides that are frequently found in foodstuffs, and one of them is indeed glyphosate. And through these types of experiments, we hope to get a more realistic appraisal or insight as to the health consequences that humans face from exposure to the pesticides, different kinds of pesticides, herbicides, insecticides, fungicides, through the food chain. So you're right, it is important not to put the blame all of our problems on glyphosate. Far from it. It's a mixture problem, and we need to look more than that.
Right.
I think what our study has shown, however, is that first of all, that doses of glyphosate Roundup that regulators have considered as having no health effect, and upon which the acceptable daily intake, the safe daily dose, is calculated, that our results are questioning the validity of the regulatory permitted daily intake, daily ingestion of glyphosate. That's the first thing. And secondly, it's really flagging up, highlighting yet again that regulators need to come into the 21st century, stop dragging their feet, and come into the 21st century and embrace the types of analyses that we have done in this study, what's known as molecular profiling, molecular composition profiling analyses, collectively known as omics, OMICS. These have revolutionized, these omics, molecular profiling analysis, have revolutionized the depth and sensitivity of the measurements, the depth and sensitivity of measurements that we can make from exposures to chemical exposures. Omics analyses have revolutionized our diagnostics in medicine. They can revolutionize also the precision, accuracy, precision, sensitivity of measurements in toxicology studies. And so far, regulatory agencies around the world are very reticent to embrace the power of these technologies to help in health risk assessment of chemical pollutants and arrive at putting in place safety measures that are in the interests of public safety.
Why do you think that is? Why do you think you see that reticence?
If I'm to be kind of blunt, I cannot understand from a scientific viewpoint, given the accepted power of omics analyses, which has been embraced in just about every other area of science, in every area of science. I don't see a scientific reason why regulators are just so reluctant to embrace them and add them to the standard measures of toxicology, standard toxicology measures that they employ at the moment to arrive at a safe dose of a given chemical pollutant. I feel that there is something else behind this, non-scientific reasoning behind this. So I'm led to believe, sadly, that there have to be commercial and political reasons why they do not incorporate this. Because our prediction is that if they did embrace these analyses, they would discover, as we have begun to show, that what they consider currently a safe dose for a given chemical may not be safe after all, and they will have to revise the regulatory permitted daily intakes of chemical pollutants, pesticides included. They'll have to revise them downwards.
Right.
Now, then one is faced with another practicality. If you go to revise downwards the exposure to these chemical pollutants, pesticides in foods, you're probably going to have to reduce their usage in agriculture. So there are all these knock-on effects, and perhaps they're simply not prepared to face these kinds of complications, right, which obviously for me is just simply not right. It's not right because it's putting these other vested interests, these almost commercial vested interests, ahead of public safety, and that cannot be right.
Do you see any regulatory movement at all in Europe to embrace this more so? We don't see that really much in the US. Do you see European regulatory officials interested in this or not?
The European Food Safety Authority has been debating this topic again for years. And there is no definitive move yet to, again, embrace these molecular profiling omics technologies as part of health risk assessment. I don't know if Robin knows anything more, but I simply have not heard. Again, the EFSA, the European Food Safety Authority, is dragging its feet on this like every other regulatory agency, as far as I could tell.
Robin, is that your opinion as well?
Yeah, no, it's true that these methods are discussed often, and they are not yet translated into the guidelines. So in some cases, it's true that we know it's working, and some regulators also acknowledge that it's working, but they are not translated into the test guidelines, and so industries are not required to use them when they are testing chemicals before commercial approval.
So just to get off on another subject just slightly here, COVID, with the COVID virus, taking so many lives and affecting so many people, infecting so many people around the world, it, in some circles anyway, has drawn discussion and attention to, our overall health and our immune system health, and how does the gut microbiome play into our immune system and our ability to, to deal with viruses like COVID or viruses that might impact us in the future? Is there a connection there?
Well, some of the substances produced by the gut microbiome are absorbed into the body and indeed are immune system modulators. One class are known as short-chain fatty acids. And if there is a imbalance in the gut microbiome such that these immune system modulatory molecules, these short-chain fatty acids, are not being produced properly in sufficient quantities and in the right balance, then this could lead to an impairment. It's one contributing factor to the impairment of good immune system functioning. So yes, this is an established link between the gut biochemistry and immune system. There is good evidence for this. So yes, indeed, anything that compromises immune function, whether gut dysbiosis or lacking of other nutrients, crucial nutrients in our diet, such as vitamin D3 or zinc or vitamin C for that matter as well. These three are particularly crucial. If we're deficient in these and the gut is also in bad shape, the immune system is not going to have the strength to react appropriately to any infection, let alone SARS infection from SARS-CoV-2, which is what causes COVID-19.
Right.
So, yes, we cannot ignore the role of the gut in this area either, I believe.
Okay. So as we wrap up, just want to ask you, anything else we should say about your study? Anything I should have asked that I didn't? Any points in particular that need to be made as this rolls out on Wednesday morning, I believe, correct?
Yeah, it'll be 12:01 midday, your East Coast.
Right.
East Coast time. So it'll be whatever time, 1 hour before then, and for us it'll be just off at 5 o'clock.
Right.
So from my side, I don't think I have anything more to say. Robin, do you have anything more to add?
No, I think we covered everything.
Okay.
Yeah, I think we're quite accurate. I don't think I said anything too outrageous.
Well, I appreciate your time and your explanation for this. It's all very interesting, and with COVID, gosh, we're also worried about our health right now, and I think we need to embrace every piece of science that we can, and hope that our regulators will do so as well, right? I mean,
yes, I think for me, if I was to make a general recommendation, while there is still unknowns, our study has suggested certain health risks health risks stemming from glyphosate Roundup exposure that need to be followed up. And these are clear indications, and questioning whether the daily intake, the regulatory permitted daily intake is safe or not, until we have clear answers to these questions, fill in the knowledge gap here, then I think it's only wise that people minimize their exposure to this and other pesticides, because that only makes good common sense. So my recommendation always to people is to eat as much organic food as they possibly can, because we know organic food has far, far lower pesticide residues generally. I know there's some contamination, but generally organic foods have a much lower pesticide residue levels. So, that my general, in the meantime, as I say, while we don't know whether these things are safe or not at these daily intake levels or recommended daily intake levels, let's play safe and protect ourselves and our loved ones by favoring an organic food diet as much as possible.
All right. Well, thank you both. I appreciate it, and look forward to more science coming out from your team.
Thank you very much, Carey. Thank you for doing this. We really appreciate it.